So now we next move on to the next important raw material, and one of the very essential counterpart of the translation machinery is the tRNA. The last day we did the mRNA, we realized that the mRNA produces two important sequences to the ribosome. One is the mRNA informs the ribosome where to start. So it presents the initiation codon to the ribosome for the ribosome to understand that this is the start point of the open reading frame. And the mRNA also provides a small purine-rich sequence, the Shine-Delgado sequence, to which the ribosomal rRNA pairs. So that was the part of mRNA. Now we are coming to a tRNA. If we look into the structure of a typical tRNA, you see that a tRNA has a 5' end and a 3' end. Concentrate on the 3' end. On the 3' end, we are seeing that there are 3 nucleotides: adenine, cytosine, cytosine. Usually the tRNA molecules are between 75 to 95 ribonucleotides in length. And even though although the exact sequence varies, all tRNA have some common features. So the first common feature just now I showed you is they have a 5' to 3' at one end is the ACC. So I read it from the 5' and CCA sequence. So this is the sequence, or this is the end to which the amino acids will be attached to the tRNA by the enzyme amino acid tRNA synthetase. Okay. Apart from that, it is also known that tRNA has some unusual bases in their primary structure. For example, it has pseudouridine, it has dihydroxyuridine, it has hypoxanthine, thymine, and methylguanine. We're not getting into the details. When we look at the three-dimensional structure of the mRNA, as you can see that there are loops in the mRNA and they are named accordingly. For example, the Xi-U loop is named because of the characteristic presence of the Xi-U in the loop. So we'll not be getting into the details of the loop because we are concentrating on translation. So let us concentrate on that particular important loop that plays an important role during the translation machinery, that is the anticodon loop. And as the name suggests, it contains the anticodon, which is a 3 nucleotide long decoding element that is responsible for recognizing the codons present on the mRNA. So it's by base pairing with the mRNA. Keep it in mind that the direction of the anticodon in the 5', 3' is this, okay? So this is the 3' end, this is the 5' end. And when we talked about the Ubel's hypothesis, I had showed you how the codon and the anticodon pairs and what is the direction that is maintained by the codon and the direction maintained by the anticodon. So if the mRNA is sitting in a 5', 3' orientation, we will have an anticodon loop. So we'll have we'll be having an anticodon loop. So we'll have the 5' end this side and the 3' end this side. So keep that direction in mind. So that is how the codon anticodon pairing will occur when the tRNA will be decoding the message of the mRNA within the ribosome. Now coming to a picture, let's go to the next slide. Before further going into the certain structural aspects of tRNA, I want to talk about this particular enzyme known as amino acid tRNA synthetase. This amino acid tRNA synthetase is an enzyme that attaches the amino acid to the 3' end of the tRNA. Now, in nature, we had discussed it in the first day of our genetic code class, is that there are 20 amino acids. So if there are 20 amino acids, we have, sorry, where did my cursor go? We have 20 different amino acid tRNA synthetase enzyme, one for each amino acid. So that is constant. So if we have 20 amino acids, then we have 20 different amino acid synthetases. Now let us look at an example. For example, I'm losing my curse again and again. For example, if we have a codon like this, A, C, U, A, C, C, A, C, U, C, A, and A, C, G. Okay. Now you can see these are 4 different combinations that I have drawn here. And these all combinations code for the amino acid threonine. Okay. Now, as I said, there is only one amino acid tRNA synthetase for charging the tRNA of threonine. So that is a dedicated one. So if there are 20 amino acids, there are 20 amino acid tRNA synthetase. Now, what does this enzyme do? This enzyme helps in charging the tRNA with the corresponding amino acid. Now, if there are 4 combinations to this particular threonine amino acid, this particular enzyme is capable of identifying these 4 combinations and charging the tRNA with the same threonine molecule. So when a tRNA is charged with the same amino acid for 4 different combinations, those are known as iso-accepting tRNAs. So once more, there are 4 combinations, 4 different combinations. All combinations will indicate that the threonine amino acid will be charged to the tRNA. So the tRNA and the enzyme system is capable of recognizing the 4 combinations and is capable of charging the tRNA with the threonine amino acid only. So the tRNA is capable of recognizing the 4 combinations and being charged with the same amino acid threonine. This set of tRNAs are known as iso-accepting tRNA. So one thing we understood that there are 20 tRNAs and there are 20 amino acid tRNA synthetase, and one tRNA can recognize more than one combinations. This phenomenon, as we know, is called the degeneracy of the code, and the process by which it is done is known as Rubel's hypothesis. The tRNA recognizing all the 4 combinations, as in this case with threonine I showed, are known as iso-accepting tRNAs. Now let us come into this mechanism by which the amino acid is being added to the 3' end of the tRNA. As you can see, this is the enzyme, and there are 2 active sites, one for binding with the amino acid and the other for binding with the tRNA. Now, when this enzyme acts or charges a tRNA with this cognate amino acid, we have to keep it in mind that this is done in an ATP dependent manner. In the step 1, as you can see, this is an example which has been shown by charging the tRNA of valine with the amino acid valine. So this is an example where we will see how valine is being added to the 3' end of the tRNA for valine, right? So the step 1, this process is known as adenylation. Again, once more, the pronunciation is adenylation. So the step 1 of this adenylation is that the amino acids, as you can see, reacts with the ATP to become adenylated with the release of the pyrophosphates. So the first step is the valine being added to the added to the amino acid, and 2 pyrophosphates are liberated. Now the enzyme does a coupling reaction, and as a result of the coupling reaction, we are seeing that the 2 phosphates are being liberated, and valine is now having an adenosyl monophosphate attached to it. Okay. The next part, the step 2. So this is the first part. So this comprises the first part, that valine and ATP are the tRNA synthesis enzyme gets bonded to valine and ATP, and there is a coupling reaction where the two pyrophosphates are broken down and released, and the energy is utilized for addition of valine to the adenosine monophosphate, a step known as adenylation. In the next step, what we see, so we are now having an adenylated adenosine monophosphate enzyme. Now this enzyme binds to an uncharged tRNA having the anticodon for the valine amino acid. So the enzyme binds with the uncharged tRNA, and as you can see, the enzyme transfers the amino acid from the amino acid adenosine monophosphate to the 3' end of the tRNA. So the enzyme is now transferred from AMP to the 3' acceptor arm of the tRNA. And now we get an amino acylated tRNA, and the adenosine monophosphate is released. So in the next step, we are seeing that the adenosine monophosphate is released, and we are getting an charged amino acyl tRNA. So the tRNA has been attached to the 3', the amino acid has been attached to the 3' end of the tRNA. So when we talk about charging, there are two steps. The first step is, as I said, step 1 is adenylation, in which the amino acid reacts with ATP to become adenylated with the release of pyrophosphate. As a result of this step, the amino acid is attached to the adenylic acid of the adenosine monophosphate by a high energy ester bond. Step 2 is the tRNA charging, which we saw, in which the adenylated amino acid remained tightly bonded to the synthetase, reacts with the tRNA. And finally, the reaction results in the transfer of the amino acid from the 3' end, transfer of the amino acid to the 3' end of the tRNA with the release of AMP. So this portion is step 1. I've again lost my cursor. So this portion is step 1, adenylation, and this portion is step, so this is 1, where we are having adenylation, and this portion is step 2, where we are having the charging of tRNA. So this is how tRNA molecules are charged by their cognate amino acid, and the enzyme responsible in the process is the amino acid tRNA synthetase. And as you can see, now the tRNA is the amino acid tRNA. Okay. When the charging is occurring, this is a bit of showing how. So this is now first let's identify the picture. You can say this is the tRNA, this is the 3' end. So we have a C, we are having a C, and this is our adenine. To this adenine is attached, as you can see, the amino acid. You all are aware of the fact that the amino acid has an amino terminal group and a carboxy terminal group. So you can see is the carboxy terminal group that has been joined to the adenine group. The carboxyl terminal group is attached to the ribose of the last ribose of the ribose, I mean to say CA. So it's attached to the carboxyl group of the ribose of the last ribonucleotide of the tRNA chain. If we look into the position numbering of the molecules within this ribose sugar, we all know that this is the 1' position because O is the functional group here. If you remember your organic chemistry, this is the 2' position which has a OH attached to it. This is a 3' position. This is a 4' position, and the carbon here is the 5' position. So this is a 5-carbon sugar, right? If you remember, you can see in this particular case, the amino group has been attached to the 3' OH of the tRNA. So this attachment has taken place in the 3' OH of the tRNA. Based on whether the amino acid is attached to the 3' end or whether it is attached to the 2' end, amino acid tRNA synthetase are of two categories. Class 1 enzyme attaches the amino group to the 2' end. So if the amino acid would have been attached here, then the amino acid synthetase belongs to the class 1 enzyme. But in this particular picture, you are seeing that the amino acid has been attached to the 3' end. So the enzyme here that was involved in attaching the amino acid to the cognate tRNA is the class 2 enzyme. Okay. So as I said, so this is the two classes of enzyme. There is one picture. I will not get into the details of the chemistry, but you can see here. So this is the adenine. So you can see the amino acid has been attached to the 2' end. And this is again the adenine. And you can see the amino acid has been attached to the 3' OH. So depending on whether it is attached to the 2' OH, the synthetase is known as class 1 enzyme. And whether the amino acid is attached to the 3' OH, the synthetase is known as class 2 enzyme. Now, let's get back to the previous slide. As I said, that most organisms have 20 different tRNA synthetase. And one more thing, concept I said that each of the 20 amino acid, this is very important, keep this in mind, is attached to the appropriate tRNA by a single dedicated tRNA synthetase. And with the example of threonine, I explained that most amino acids are specified by more than one codon. So it's not uncommon for one synthetase to recognize and charge more than one tRNA. So the 4 sets of codons of threonine was being recognized by a single tRNA, and these are known as iso-accepting tRNAs. This is the concept I have already given you. There's one more thing I would like to mention here. At times, how do we write a tRNA? For example, if we write a tRNA as methionine, and let me give you an example here, methionine dot tRNA, and on the superscript we write methionine. Keep this in mind that this portion of the nomenclature, there's a dot here, okay? This portion of the nomenclature indicates that this tRNA is for methionine. And this portion of the nomenclature, and this portion of the nomenclature indicates that to the tRNA for methionine, methionine amino acid has been added. So you can see another nomenclature given here in this picture. See, so it's tRNA for glutamine. And had glutamine been written in front, then we have to assume that glutamine has already been added to the tRNA for glutamine. So when we write this nomenclature, we say that the tRNA is uncharged, but the tRNA is for glutamine. And when we use this kind of nomenclature, then we have to say that the tRNA is charged. That is, the tRNA for methionine has been charged with methionine. I've drawn a picture to compare the two difference between the two nomenclatures. In this picture, as you can see, I've drawn two tRNAs. So this is the uncharged tRNA for serine, bearing the anticodon, which will recognize the codons of serine. This is the uncharged tRNA for methionine, bearing the anticodon, which will recognize the codon for methionine. And now look at here. I've shown another tRNA with a slightly different nomenclature. You can see that this is the uncharged tRNA for methionine. And when we compare this picture with this one, you can see methionine is sitting in front of it, which indicates that the tRNA for methionine has been charged with the amino acid methionine. So this is the nomenclature for a charged tRNA, and this is usually the nomenclature for an uncharged tRNA. And finally, so once we have done that part where amino acid synthetase recognizes the iso-accepting tRNAs and accordingly charges the tRNAs, with the respective cognate amino acids. So it's very it's very much understandable that the amino acid tRNA synthetase, there are 20 amino acid tRNA synthetase, and this 20 amino acid tRNA synthetase are specific for the 20 amino acids. So there might be a recognition system by which the enzyme recognizes a particular amino acid as well as the tRNA that accept that amino acid. So see, the enzyme must recognize two things. One, for example, it is we studied the example where the amino acid charged the tRNA with valine. So it has to recognize the amino acid valine. It has to recognize the tRNA for the amino acid valine. If you remember the enzymatic pathway by which amino acid synthetase works, just now we did that. You remember that the enzyme first binds with the amino acid. Then, so first step is the adenylation. So that is occurring first. And once that is done, step 2 is that it binds with the tRNA and then accordingly charges the tRNA with the amino acid. So the main important part for that enzyme is to recognize a particular amino acid as well as a tRNA that accept that amino acid. So how does this amino acid synthetase in the enzyme do that? It has been observed that the recognition of the appropriate amino acids has been based on different size of the amino acid, charges of the amino acid, and the R groups of the amino acid. So when it recognizes an amino acid, it is based on the charges of the different groups, different size of the amino acid, the charges present on the amino acid, and the R groups present on the amino acids. So this is how it recognizes an amino acid. How does it recognize a tRNA? It has been seen that there are certain nucleotides which are important in recognition of a particular tRNA. So this picture shows the nucleotides. If you look into the tRNA structure, these three sequences, C, 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 A, that's it. C, C, A, that's constant. And this sequence cannot be used to differentiate between the tRNAs. So the enzyme does not use to differentiate these between the tRNAs. This is a constant structure. Now, if we look into the part where we are having certain yellow positions, look, these are some nucleotide sequences. These nucleotide sequences are also recognized by more than one synthetase. Now coming to the red portions. So 1, 2, we are having some red nucleotide sequences, which are also known as discriminator bases. These bases, so this red color bases, these bases helps in discriminating between the tRNAs for a particular tRNA synthetase. So these positions in red are important in recognition of the tRNAs by one synthetase. So these bases, as I said, are known as discriminator bases and as referred to as the second genetic code in the book of Watson's Molecular Biology. So as per Watson in his book, he states that there are two genetic codes. One is the first genetic code. First genetic code is the codons present on the mRNA, which actually carries the information as to which amino acids will be added in a string to give rise to a polypeptide. So that is the first genetic code. And according to that book, there is also a second genetic code. The second genetic code are those red dots on the tRNA, which helps the amino acid tRNA synthetase to recognize a specific tRNA for a specific amino acid. So that this concept is only present in the book of Molecular Biology of Gene by Watson. So while studying tRNA, we developed the concept of the first genetic code and the second genetic code. And we also developed the concept that there are two categories of amino acid tRNA synthetase, class 1 and class 2, depending on to which OH they are adding the amino acid of the adenine molecule. And in the mechanism of charging, we studied there are two steps of charging. One is adenylation, in which ATP is attached to a amino acid. And the second part is the tRNA charging, where the amino acid is then added to the cognate tRNA. Now coming to the last raw material of translation machinery, before we can move on to translation, are the ribosomes. As you can see, I have given you a schematic representation of the ribosomal subunits present in prokaryotes, that's the bacterial ribosomal subunit, and the one in the eukaryotes. This is a schematic picture. For your syllabus, we will restrict ourselves to the prokaryotic system. However, if you compare the ribosomal subunits, in both prokaryotes and eukaryotes, ribosome consists of two unequally sized subunits, though as you can see, the large subunit, this is the large subunit, this is the larger subunit for eukaryotes, and we have a smaller subunit, this is the smaller subunits for eukaryotes, and this one is for prokaryotes. Each subunits, as you can see, contains one or more ribosomal rRNA molecules. So these are, for the larger subunit, these are the two categories of ribosomal rRNA molecule. And for the smaller subunit in prokaryotes, this is the only one ribosomal rRNA found here. And if you remember, the 16S ribosomal rRNA plays a very important role in recognizing the Shine-Dalgarno sequence in the 5' leader region of the mRNA. And the ribosome, as you can see, it is made up of RNA molecules and proteins. In case of the 50S subunits, we are having the 23S ribosomal rRNA. We have the 5S ribosomal RNA and 31 proteins. When you look into the smaller subunit, we have the 16S ribosomal RNA and the 21 proteins. So we get a similar picture in case of the eukaryotes. And as there is one more thing I want to show here is that during translation, as you can see, the mRNA, you can see the mRNA is passing through the ribosome, isn't it? So during translation, as you can see by this particular picture, the mRNA actually passes through the small subunit of ribosome. This is something very important. It is passing through the small subunit of ribosome. That is how I've showed you the direction. And the ribosome moves in this direction. The ribosome is moving in this direction. And the mRNA is passing in this direction. And as you can see, specific sites on the ribosome are present that binds tRNA at different stages of polypeptide synthesis. So we have a E site, which is the exit site. We have a P site, which is the peptidyl site. And we have an A site, which is the amino acid site. Now, the amino acid site is the site where an incoming amino acid tRNA binds. A fresh new amino acid tRNA comes and binds here based on the codons present here, okay? We have the P site where the tRNA is carrying the growing polypeptide chain. We'll be getting into details of this sites later on. And we have the E site where the tRNA binds. The tRNA binds on its, the tRNA that was present in the P site, when will be leaving the ribosome, will enter the exit site, and then the tRNA will leave the ribosome. So as you can see, a new tRNA enters the A site. Ribosome moves 3 units. This tRNA moves to the P site. And after its amino acid has been attached to the growing polypeptide chain, the tRNA finally moves to the exit site, again when the ribosome moves 3 units, and the tRNA leaves the translation machinery. So this was in nutshell, till date what we did the mRNA, tRNA, and the ribosomes, the 3 important raw materials essential for translation, who come together and in a very coordinated manner and in a very efficient manner also decode the information present on the mRNA to produce a corresponding polypeptide. So we'll stop our class here today. We'll start with the initiation process the next day.